Though only one partner hospital, the Istituto Giannina Gaslini in Genoa, was involved in the work on neuro-oncology, Helfischal developed a study still based on the principle of data integration, and so applicable to many, many more hospitals in the future. Let's listen to Maria Luisa Garret, our lead oncologist from the Istituto Giannina Gaslini, explain the design of the project.
Childhood brain tumors are disease that affect brain representing more than one tumor, more than one disease, and the diagnosis and treatment requires the expertise of many components, from neurosurgery, neuroradiologists, pathologists for diagnosis, oncologists, radiation therapists, and etc. So our aim is to integrate, to create a database that contains all the clinical data, surgical data, imaging, neuropathological data, and staging results, and the results of the biological study.
Clinically, we wanted to exploit the integrated data to identify prognostic molecular markers which could help predict clinical outcomes. Now we can listen to Valeria Capra explain the clinical aims and how this integrated data was used.
These are, during the years, many genes has been identified as possible, having possible roles in different histology of gliomas. This project has had a big integration of first the clinical information that she gave us, and obviously the interaction of pathology, neuroradiology, and we want to add also some molecular information. So the clinical aims was to understand the pathogenesis and finding prognostic molecular markers association with clinical and genetics, and that could predict the clinical outcomes. And obviously our final aims will be to improving clinical workflows. This is the methodology that we applied. We used the chip arrays from Affymetrix. We did, obviously, this process started from the histological assessment, and the tumor cells and the tumor that we used were only the tumors that had the content of at least 80% of tumor cells. We verified the diagnosis and the grading. And then from those specimens, we extract RNA. We did the quality control. We processed the RNA according to the Affymetrix protocols, and we did the prioritization of the chips. The statistical analysis was done by Siemens and by Alessandro's group.
Alessandro Verri, from the Department of Information Science at the University of Genoa, will now tell us how data-driven statistical tools and a priori knowledge were used to whittle down 54,000 genes to the few dozens that clinicians expected to be related to the disease.
So we're dealing with, at least from my viewpoint, these are a problem we'd like to address from a statistical viewpoint or from an inferential viewpoint. So we're particularly interested in figures. And speaking of data living in roughly 54,000 dimensional space, those are the figures we are dealing with. So those are the points. And this is the pipeline. And we've been lucky enough in this project to be able to start with a relatively large set of samples. As we heard from Maria Luisa, I mean, this is a relatively large, or possibly as large as it could be, sample of brain tumor. And the gene expression coming from the Affymetrix platform. And so we got into at this level and a little bit at the level 5. But basically, most of our effort was done at level 3. So I like to think that this is well, we like to think that this is a difficult problem. And the way we approach the problem, at least in Genova, is from the point of view of what we call the rigorizing way of dealing with these huge dimensional problems using sparsity as the driving force. So this sparsity in this case is the prejudice we have about this problem. There are 54,000 gene expression, or actual probe set, I say it out of the, but a huge number of expression, and we believe, and the doctors believe, that only a few, or a relatively small number, or fraction of genes are actually involved in the disease. So we're going to use this prior knowledge. So we don't know which one are, but we know that there are not 54,000 definitely. And so we have to go through a procedure able to select a few of those, a small fraction of those expressions. We set up a careful, bias-free framework in which we can actually select the model and do all the things in a way that we are sort of reassured that despite the fact we are working with a formidable problem, we can provide some significant statistical significance to the list we suggest to the clinicians and to the biologists. And this is the results we got. So this is one example. So this is now history in a sense. So this is what we gave to Gaslini a while ago before the real-time PCR. And this is a low-dimensional plot to show that, well, there's still a few genes, but still at such a large space we cannot plot more than 3 at a time. So this is a low-dimensional projection. So the blue and the red are quite separated. We like to think that what we did is just the first step. We didn't use, in no sense, and actually this was complementary to what the Siemens folks was doing. We didn't use in no sense what is available in biobanks, for example, or textbook or whatever, what I like to call the digital, what could be called the digital libraries knowledge. So our approach was definitely only data-driven. And I think, of course, I mean, there is not much more you can extract out of those data. And the interesting thing to explore currently in the near future is the interplay in between what is available out there and these statistical techniques. And the good news is that there are already a few ways we can actually explore in order to inject this prior knowledge into in our methods. And specifically, for those of you who are aware, is like kernel design, or the possibility, for example, of, as we're working on right now, to develop nonlinear feature selection techniques.